talk that is given by Marina Vivarelli from Rome on multi-drug resistant nephrotic syndrome. Marina trained in pediatrics at the University of Pavia in Italy. She completed a research fellowship at Harvard, Children's Hospital Boston, studying neural B cell function. And since 2006, she's in the Division of Pediatric Pathology at Bambino Gesù in Rome. She's worked to establish a new front of translational research in pediatric immune-mediated renal diseases and the popular nephrotic syndrome.
So this is forward.
Yeah.
Yes. Okay, perfect. Okay.
Okay, so thank you very much for the kind invitation. First of all, I'd like to thank France. I'm very excited to be part of this absolutely staggeringly interesting meeting. And so we're going to completely switch gears now and move to diseases in which there is not a monogenic cause and the pathophysiology is by and large unknown. So we have a completely different approach to finding treatments for these diseases. So first of all, I'd like to sort of talk a little bit more in detail about exactly which types of patients we're going to be trying to target and treat. So we all know that idiopathic nephrotic syndrome in children is a world extremely heterogeneous disease. A large percentage of these children are steroid sensitive. Luckily, there is a percentage of children who are steroid resistant, about 10 to 25%. But of these, about half, sometimes even a little bit more, respond to second-line immunosuppression, so calcineurin inhibitors. And both these patients and this initially steroid-sensitive nephrotic syndrome patients then progress to have a disease which can last for decades with multiple relapses, and in a certain percentage of cases also last into adulthood. The multi-drug resistant nephrotic syndrome patients, the patients who don't respond to steroids and don't respond to a second line, which usually is a calcineurin inhibitor, are called multi-drug resistant nephrotic syndrome. And of these, we know that a large part are genetic forms, and these will never respond to immunosuppression because they have a monogenic disease of the podocyte, which will be probably addressed by the speaker after me, Moin Saleem. And these diseases invariably lead to end-stage renal disease. However, they do not recur post-transplant. Instead, the multi-drug resistant nephrotic syndrome forms have an immune-mediated pathophysiology, which we don't understand. So they're particularly tough to treat. And not only can they lead to end-stage renal disease, but they also tend to recur following renal transplantation. So the kids that I'm going to address as our problem of the current talk today are these. So these are the patients that have a protracted, prolonged course of nephrotic syndrome with the need for immunosuppressive therapy for a long time. Some of them will not reach end-stage renal disease. Some of them will. And the ones who do tend to recur post-renal transplantation. So I'm going to go through some clinical scenarios just to drive this concept a little bit closer, and I will then try to go back to these clinical scenarios at the end of my talk and give some solutions. So one of the scenarios is the primary multi-drug resistant child. So this can be, for example, a child who has an onset of nephrotic syndrome quite late. So when you do not suspect a genetic cause, around 9 years of age, for example, he doesn't respond to the 6 weeks of oral prednisone and to 3 pulses of IV methylprednisolone. And so he's defined as steroid resistant. He also doesn't respond to the second line, a calcineurin inhibitor. So he becomes defined as a multi-drug resistant nephrotic syndrome child. And he can go through, for example, 2 cycles of rituximab. He has a negative genetic panel for all the genes coding for podocyte proteins. And histologically, classically, the first renal biopsy, which according to guidelines is usually performed at 4 weeks, will show often minimal change disease. However, if you repeat the biopsy further down the line, even to see how much margin you have to continue treating this patient, you almost invariably will find that he or she has progressed to focal segmental glomerulosclerosis. These children often have a rapid progression. And for example, in the child that I chose to describe, 15 months after disease onset, he proceeded to end-stage renal disease. And we know that this child, if he doesn't have a genetic mutation or a variant, a pathogenic variant, has a substantial risk, in this case, around 30 to 50% of recurring following renal transplantation. The second scenario is a completely different scenario where we don't get to end-stage renal disease. These are the children who maintain steroid sensitivity. So for example, a child with a nephrotic syndrome onset at 4 years, response in 3 weeks. So he's a steroid-sensitive nephrotic syndrome because he's responded within 4 weeks. He then proceeds to have multiple relapses over the years. Some of them are steroid resistant because they take more than 4 weeks to respond and or because they require IV methylprednisolone. In this case that I chose to describe, this patient, which I then saw for the first time when he was 16 and a half, had never achieved stable remission for at least 12 months, despite having used for prolonged periods of time CNIs, MMF, mycophenolate mofetil, multiple infusions of anti-CD20 monoclonal antibodies, both rituximab and ofatumumab. He had stunted growth, osteopenia due to prolonged use of glucocorticoids, and signs of a CNI toxicity. So yes, he had a preserved renal function, but he had a very severe load of toxicity and morbidity associated with his primary disease. Another clinical scenario, and this is the last one, is so-called secondary steroid-resistant nephrotic syndrome. So for example, a child with an onset at 7 years of age who is a late responder. So he responds in the window between 4 and 6 weeks of oral prednisone. Subsequently, though, he did achieve response, and then he was continued to treat with 3 pulses of IV methylprednisolone and CNI. He had subsequently multiple relapses, some of them steroid resistant, taking more than 4 weeks to respond. He had use of CNIs, MMF, multiple infusions of anti-CD20 monoclonal antibodies, so stunted growth, osteopenia due to prolonged use of glucocorticoids. However, to make it worse, in addition to what I presented in the previous patient, this child, 2 years following disease onset, had a multi-drug resistant relapse. So he relapsed nothing that we could do for him, including plasmapheresis-induced remission. So this child progressed to end-stage renal disease. So these children, the secondary steroid-resistant nephrotic syndrome, as Moin Saleem's group has showed very convincingly, are the ones who have the truly highest chance of recurring post-renal transplantation. So secondary steroid-resistant nephrotic syndrome leads to end-stage renal disease with a negative genetic panel. This child was mononephrectomized, put on peritoneal dialysis for about 6 months, and then received renal transplantation from a living donor. And as was predicted by his chances, given his clinical history, he recurred almost immediately following the renal transplant. And this was also histologically documented. So at this point, also here, he was treated with intensive immunosuppression and also plasmapheresis. And I'll go back to his treatment further on at the end of my discussion. So we know that idiopathic nephrotic syndrome in children can be a long and bumpy road, extremely heterogeneous, and to define the best treatment strategy, we would need to understand the physiopathology. Unfortunately, we don't. And I think this is, in stark contrast to all the different diseases who have been presented throughout the course of this day, unfortunately. So we know that there's something triggering dysregulation of the immune system, but other than knowing that there are trigger events who lead to a disease relapse, we don't have a very clear understanding of what's going on within the immune system to go to proceed to damage the podocytes and induce proteinuria. So there have been different ways and approaches to enhance our understanding of this. One has been to look at genome-wide association studies. And there have been different studies that have been performed over the last few years. Here I present results from the 3 main ones that have been published in the last 10 years. And these have given us a very important response because they have showed us that there are different genetic variants predisposing to developing steroid-sensitive nephrotic syndrome that are in the HLA locus. So that SSNS is associated with the HLA region and in proteins encoding for MHC class II molecules. So clearly an involvement in the interaction between B cells and T cells and T cell co-stimulation. Further reinforcing the fact that these are diseases that are driven by an immune dysregulation. This is a nice review that I'm just showing to you. I'm not going to go through the results of all the different GWAS studies that have been performed up to now. It was published this year in Pediatric Nephrology, and it shows both the HLE SNPs and also SNPs that have been found outside of the HLE region, most interesting one, for example, in nephrin. And I think that this approach is an approach that will allow us moving forward, not only to identify potential mechanisms and therefore potential therapeutic targets, but also to cluster this extremely heterogeneous group of patients into patients with a more similar phenotype in terms of time of presentation, modality of prognosis. So I do have the fortune of being able to very briefly show you also unpublished results from the most recent and most important up to now in terms of numbers, genome-wide association study to which many of us also have been able to participate, which has been led by Matt Sampson at Children's Hospital Boston, and has been able to collect 2,440 cases from all over the world. And he has presented these results at IPNA, so I'm not going to go through them in detail. But he has been able to reproduce some of the previously identified SNPs and also to add a few new ones. And I think, again, that this type of an approach will be able to help us define our targets and define our subpopulations moving forward within this extremely heterogeneous disease. So moving on from lessons learned from GWAS to lessons learned from clinical efficacy, and so from the therapeutics that we use for this disease. So the most useful indirect evidence that has helped us move forward in treating these children has come from the efficacy, completely unexpected, of rituximab. So the anti-CD20 monoclonal antibody that depletes CD20 positive B cells. And this is the definitive trial published by EGM and The Lancet. And this is just something showing from a cohort of 40 patients followed in our hospital, how this is the only therapeutic agent up to now who can be disease modifying. So there have been children treated with rituximab, even with a single infusion, but never relapsed again, even looking at them 5 years down the line. So this clearly indicates that we're doing something right when we're giving this type of a drug, but we don't know why. So rituximab and also ofatumumab, the humanized form of an anti-CD20, has an effect on depleting B cells. And by depleting B cells, it also depletes, because they're its precursors, short-lived plasma cells. This is shown clearly here, where we had the opportunity to compare rituximab and ofatumumab, and we found that the effect on all the B cell subpopulations was similar at 1 month, and also on CD20 negative plasmablasts, as I showed in the previous slide. So we just reconfirmed this, and we didn't find a difference between ofatumumab and rituximab in terms of B cell depletion. We also looked at the different subpopulations with these two drugs, and what we found was that these drugs are able to very effectively and for a prolonged period of time deplete memory B cells. And why do we think that depletion of memory B cells is particularly important? We think that this is important because we and others have shown that memory B cells are able to predict relapse following anti-CD20 treatment. And this was initially shown by our group in a small cohort in 2016, and has been where we showed that this percentage of memory B cells was highly predictive of relapse. And this was subsequently reconfirmed in a different cohort, so with a completely different ethnicity in the Indians by Arvind Baga's group, and also by Paulo Cavedi at Columbia. We also have unpublished data now showing that memory B cell levels at baseline, so before giving anti-CD20, can predict relapse, and that children having higher levels of memory B cells are much more likely to relapse. So this can be something that can allow us to fingerprint our patients moving forward and to choose who to treat and when. So we know that B cell depleting treatment has a large variety of effects on B and T cells. And I'm going to show you more in depth and more in detail the evidence that we have for a role of B cells in the pathogenesis of nephrotic syndrome. So this is just an overview figure, and I'm going to go through the different parts of it. So in terms of B cell phenotype in children, moving backwards, so based on our observation of the effect of rituximab in these children, we tried to look at a B cell phenotype at disease onset in these kids. And what we confirmed was that even at disease onset, total B cells are higher in children with steroid-sensitive nephrotic syndrome before receiving any other form of treatment. And also the different subclasses of B cells are higher. But particularly what we saw is that with treatment, all the different subclasses go down, So you see that transitional, mature, and total B cells go down. The only one that instead goes up are memory B cells. And we think that really this is, again, an indirect sign of the efficacy and the importance of the pathophysiology of this subgroup of cells that instead remain lower in remission. So in adults, is this also the case? So there aren't many studies in adults, and we're trying to address this in minimal change disease in adults. But from the results that we have, it's not like that. Memory B cells do not seem to be important in adults, where instead there's an increase in plasmablasts. And if you think about the way in which adults respond to prednisone at disease onset, minimal change disease, it really does seem to be a different disease. So we think there's something very different between the pathophysiology of this disease in children and in adults. And the initial data in B cells seems to confirm this. So when we look at pathogenic antibodies, there's been a lot of evidence looking at different types of pathogenic antibodies in nephrotic syndrome. This obviously it brings more value to the hypothesis that B cells are important in nephrotic syndrome. So one of the experiments that has been done and was actually done by our group and looked at hyposialylated IgM and its role in nephrotic syndrome. And this stemmed from an initial observation in which we saw that a subset of children with INS at onset, so before receiving any immunosuppressive therapy, had very high levels of IgM on the surface of their T cells. And these were the children who did more poorly, who relapsed sooner. So they seem to identify a subgroup of steroid-dependent patients. And when we looked at patients in different moments of the disease, we confirmed this. The steroid-dependent nephrotic syndrome children have higher levels of hyposialylated IgM on the surface of their T cells. So why is this important? This took us quite a lot of time to try to understand. So pentameric IgM is sialylated in a whole variety of spots. And the sialylation of pentameric IgM helps it bind, and these are natural IgM, to the surface of T cells and actually inhibits the T cell proliferation. If the IgM are desialylated, and these patients do have desialylated IgM, as we showed here, these have a reduced capacity of proliferation, of inhibiting proliferation of T cells. And this we did performing experiments using commercially available IgM, and we showed it here, where we showed that the desialylated IgM are proliferate just as much as T cells that do not have IgM on the surface, whereas when they have sialylated IgM, they proliferate much less. And this is also connected to the loss of inhibitory capacity of dexamethasone. These are in vitro results, of course, but in vitro, these cells are less susceptible to inhibition of dexamethasone proliferation if they have desialylated IgM on their cell surface. So other pathogenic antibodies have been identified. There's been a flurry of interesting experiments and results in the last few years. And the three main antigens that have been identified are CD40, UCHL1, and nephrin. So these are the three papers that have been published. In nephrin, anti-CD40 antibodies were shown in adults in a cohort of relapsing FSGS patients by Delville. And this was also shown, these antibodies were shown to be pathogenic in mice. In children, Georges Deschamps' group found the existence of anti-UCHL1 antibodies, and again showed that these were pathogenic in a mouse model. And much more recently, both in children and in adults, Astrid Vine's group at MGH showed the presence of anti-nephrin antibodies, which were particularly interesting and also quite fascinating, given also the GWAS results in minimal change disease, both in adults and in children, both in the circulation and in renal biopsies. Another aspect of podocytes in which they can act as antigen-presenting cells, which is hotly debated, but I thought I would mention it because it does have therapeutic implications, is that the fact that in some situations, injured podocytes appear to be able to act as antigen-presenting cells by expressing B7-1 or CD80. So this is hotly debated because this was first shown by Jochen Reiser's group in 2004, where he showed that murine podocyte upregulation of B7-1 induced proteinuria. However, when almost 10 years later, this was brought into the clinic with the abatacept paper that was published in the New England Journal, abatacept was shown to be effective in 5 FSGS patients. And abatacept, excuse me for not mentioning this, is a fusion protein that is composed of unifying IgG1 with CTLA4 and binds CD80. And this monoclonal antibody appeared to be effective in a small group of patients, even though they were patients that had received a lot of stuff at the same time. So it was not very easy to dissect out the positive effect of abatacept. More importantly, these patients seemed to express B7-1 in their renal biopsies. However, this was not reconfirmed subsequently. On our side of the pond, in a European cohort, B7-1 was not expressed in patients with FSGS, and this was also looked at by Giuseppe de Mossi's group in Bergamo and also not confirmed. And also, prospective trial of 9 patients with FSGS with a relapse post-transplant treated with abatacept did not show any response. However, more recently, again, the Americans have showed another positive cohort response in which they treated 12 patients, many of them children, 9 out of 12 were children, and they showed that in the cohort of patients who did express B7-1, there was a clinical response to abatacept. Of these 9 patients, 6 had positive B7-1 in their renal biopsies. The ones who did not either had not had a biopsy performed or had a biopsy performed before reperfusion, so it wasn't informative because they didn't have the relapse of disease anyhow. So I'm not sure that this is data that will be able to be reconfirmed, but I think that given the absence of alternative therapeutic options, it's something that we need to keep in mind. So the fourth and last thing that I wanted to discuss about is antibody-independent effects on B cells that might also have an effect on podocytes. And on this, I just briefly wanted to mention BAFF. BAFF is a B cell survival cytokine. And we thought that BAFF might be useful because there is a monoclonal antibody targeting it called belimumab. There are also others. But we thought that given the fact that this had been shown to be useful also in membranous nephropathy, it might be useful in frequently relapsing nephrotic syndrome. This is a very small pilot study that was performed and that did not show a significant effect. However, I do not think that belimumab should be ruled out just because of this initial observation. I think that if we think about the importance that B cells seem to have in these forms, difficult forms of nephrotic syndrome, and if we think that when we're giving a depleting agent such as anti-CD20, we're then getting an increase in BAFF to reconstitute the B cells, at that point, giving BAFF might be useful. This has been done in lupus nephritis. So you give rituximab, and then when the BAFF goes up, you give belimumab and prolong the effect of the anti-CD20. And this is something that could probably quite safely be used also for nephrotic syndrome. So going back to the clinical scenarios, I'm going to go back to the severe steroid-dependent child that I had showed you before. We have all encountered in our clinical practice this type of a child with all these relapses, all this toxicity, normal renal function, but a really, impaired quality of life. What do we do in these patients? So one approach which has been done by Claire Dossier in Paris, which I think is innovative and interesting, but quite, with a significant toxicity, is to give obinutuzumab, which is a new anti-CD20 monoclonal antibody, a class 2, which has the ability to induce a more prolonged and protracted B cell depletion, followed by daratumumab, which is an anti-CD38, so plasma cell depleting agent. And they treated 14 patients in which they really had, no other thing to do. These children had tried everything under the sun and they had not improved. And in these children, I think I remember correctly, 9 out of 14, they had a very significant clinical response. However, they did also find the prolonged use need for IVIG for about 1.5 years, depending on the patients. But this is something that is innovative and that's probably moving in the right direction. So what about trying something that is a little bit less toxic? So we thought about trying mesenchymal stromal cells quite a few years ago, following a pilot study that we had performed, in which we thought, we know that mesenchymal stromal cells are immunomodulatory. They have 0 side effects. So let's try to see if we can purify them and produce them from these patients and use them in a safe fashion. So we had performed a pilot study in which we took bone marrow when we were doing biopsies, renal biopsies, from 5 children with really severe forms of either steroid-dependent or steroid resistant but responsive to immunosuppression children. We took a little sample of their bone marrow, we expanded it to form mesenchymal stromal cells, and we looked at their phenotype and function. And we saw that their phenotype in terms of capacity to differentiate into the different tissues that mesenchymal stromal cells produce was absolutely normal. We also looked at their immunomodulatory function in vitro, compared to healthy controls, so to mesenchymal stromal cells from healthy controls, and we saw that they were also completely normal. So based on this, we designed a phase 1 study called the MESNEF study, in which we chose to try prospectively to use autologous mesenchymal stem cells in children with very severe forms of steroid-dependent nephrotic syndrome. They would receive 2 MSC infusions at day 0 and day 8. And from a month after the first infusion, they would start tapering all the immunosuppression that they were on, and we would look at them for 12 months. Obviously, as this is a phase 1 study, the primary outcome was safety and feasibility. And this is the demographics of the study of the children and adults that we treated. Overall, we treated 10 children and 6 adults with a very long disease duration, and also having received a ton of immunosuppression previously. And very briefly, these are the results, just looking very, very simply at the number of relapses in the 12 months before and the 12 months following treatment, and the number of immunosuppressive medications before and after. And what we saw was that there's a pretty striking effect in the children and absolutely no effect in the adults. So I think this is interesting and probably points to the fact that there is really a difference in these diseases as they move forward. So the last clinical scenario that I wanted to go back to is the nightmare, the secondary steroid-resistant nephrotic syndrome child who then tends to relapse. So going back to the child that I had described before, this child promptly relapsed following his renal transplant. He actually relapsed on the operating table. As soon as his diuresis restarted, he had proteinuria. And we confirmed this histologically. We followed the classic protocol of rituximab followed by intensive plasmapheresis. However, after 4 weeks of rituximab and 12 sessions of plasmapheresis, we decided to give him 1 dose of obinutuzumab instead of rituximab. And he actually continued plasmapheresis, but achieved complete remission. So we have no idea whether the remission would have arrived even if we had given rituximab, in all frankness. However, what we were surprised by, and we usually do not see with rituximab, is the prolonged depletion. So at 6 months from OB, his B-cell depletion is still complete. He had managed to taper his plasmapheresis to 2 sessions per week, and he had non-normal renal function and UPCR, borderline normal, both of them, but satisfactory given the severity of the phenotype. And at 9 months, he's doing plasmapheresis now once a week, and he has a satisfactory UPCR of 0.48 and a stable EGFR. This is also instead a small pilot study that we are hoping to be able to implement for these particularly difficult patients within the ERKNet the grant that has been submitted. It's going to be called the Focal Step Study, and it addresses FSGS relapses post-renal transplantation or patients with severe multi-drug resistant nephrotic syndrome. And in these patients, we're interested in following a stepwise approach in which we try one approach. After 6 months, if it hasn't worked, we try another approach, and so on. And the 3 approaches that we're going to look at are abatacept only in B7-1 positive patients. We're actually curious in doing this just to see if B7-1 is actually found in our patient cohort or not. And in those who are abatacept negative, obinutuzumab for 6 months. If they have a positive response, we continue. Otherwise, we move on to daratumumab. So in conclusion, I think for this group of diseases, which are particularly challenging, we are better off now than 50 years ago, but we are far from meeting the clinical need. There is a need for collaborative efforts, both for pathogenesis and for prospective randomized control studies. Pilot studies with innovative approaches, sampling pre and post treatment are crucial. And I think there is much more information to be gained by large unbiased genomic and transcriptomic efforts. I'd like to thank all my collaborators, our funding agents, the patients, their families, and you for your attention. Thanks.
Thank you very much. That was a high one of data. Questions in the room. So So again, maybe I start. Looking at the data with memory B cells, would you consider that maybe, looking at the initial data already, you would sort of take decisions on the intensity of anti-CD20 treatment? Would that be a path to follow?
Yeah, so that's a really good question. So essentially, that's exactly the direction that our research is trying to move in. And the whole idea is to do tailored treatment. So I think everyone in their experience has, tried rituximab in a lot of these different patients. And there's some patients who respond really, really well, and others who really don't. And we really don't have a very clear idea of why that is. And that's really always been for me sort of the crux of the issue. Also because rituximab is expensive and also has quite a few side effects. And, we're not treating cancer here. We're treating something that has other therapeutic options. So trying to figure out which patients will respond is definitely where we're trying to go. And I didn't have time to show you all the data that we have, but we have clear indications now that age plays an important role and that older children will respond better to rituximab. So it's probably quite appropriate to wait and give it when they're a little bit older. They have a much higher chance of responding. And we also think that, yes, the levels of memory B cells at baseline are going to influence how well they respond. So we probably need to find something else to pull down their memory B cells, or say, okay, maybe for this child, we need to choose a different therapeutic approach.
Matthias.
Yes, so I noticed that in the GWAS low side is shown in the beginning for the steroid resistant nephrotic syndrome was NPHS1, whose mutations cause a...
Not for steroid resistant, for steroid sensitive.
Yeah, that's what I mean.
Okay.
Whose mutations cause are a classic form of steroid resistant nephrotic syndrome. So how do you interpret this? Is this a steroid resistant modifier effect, or is this related to the fact that the antibodies are actually binding to nephrin?
Yeah, so I think we were all super blessed and really surprised when nephrin started to come out in the first cohort as one of the SNPs that predicted steroid-sensitive nephrotic syndrome. But it's been confirmed in a really large cohort now, and in parallel, having Astrid Vines showing the anti-nephrin antibodies sort of brought it home. And I think that's by far the most likely explanation to that finding.
Any questions on that?
Marina, there is some interesting experience in the use of lipopheresis to treat both native but also post-transplant FSGS. And do you think that lipids could be a potential target for this condition?
Yes, I do. I think that is also something that we would probably need to acquire more experience with putting together a large group of patients, but it's just very difficult. Even there, the impression is that it doesn't work for all patients. And another thing that I have discussed also with Anya Kozil, who treated quite a few patients at the Evelina in London, was whether the LDL pheresis makes the patient more susceptible to the CNI, because achieving a good and consistently high CNI therapeutic level when you have such a important dyslipidemia is very difficult, and this may be a co-cause of the response that we see in these patients. But this is just something that we need to look at more in detail. We don't really know.
All right. Thank you very much, Marina. And then we go, as well as a leading national and international, and of leading national and international registries on nephrotic syndrome. Moin is also the founder and chief scientific advisor of Pure Spring Therapeutics, focusing on the podocyte for renal gene therapies. And apart from all these qualifications, he's a great football fan, and he's already given his opinion on yesterday's events taking place with the German body. So we're looking forward to.
Yeah, well, thank you for your extreme hospitality of Germany in particular. It's always, as an England football fan, it's always difficult to come to Germany during a major football tournament. But this time, I feel much more relaxed. Nevertheless, you're going to knock us out on penalties in the knockout stages, and let's say it's too early. So thank you. Fantastic meeting. I've really enjoyed it so far, and I hope that I can sort of continue the theme. Marina's very nicely introduced nephrotic syndromes for us, so I'm going to talk to you predominantly about the genetic forms of nephrotic syndrome. And clearly there's a huge unmet need here. There's more than 80 genes that have been described that, if mutated, cause nephrotic syndrome. And the most common of these is NPHS2, which encodes podocin. So at least 20% of the monogenic causes in Caucasian populations are caused by NPHS2 mutations. There's no current treatment. And what I want to talk to you about today is a couple of studies that have been done by very talented PhD students in my lab over the last few years that are looking at ways of addressing that. So the first, and as I say, so most of the genes, well, the genes that we all know about, they're expressed in the podocyte. The genes, in particular NPHS2, is expressed at the slit diaphragm and causes the most severe, the slit diaphragm mutations tend to cause the most severe phenotypic disease that we see in the earliest onset. So we wanted to look at podocin. Podocin was our gene of interest for these studies. Podocin, biologically, it's a podocyte-specific membrane, integral membrane protein. And mutations, as I say, the commonest one is genetic nephrotic syndrome. And the most common, most frequent mutation is R138Q, and this is a misfolding mutation that causes mistrafficking of podocin. The podocin gets stuck in the endoplasmic reticulum, and the question we wanted to ask was, can we discover small molecules that will correct this trafficking defect? So that was the hypothesis. That's what we wanted to do. Can we correct the defect? And then once you've corrected that trafficking, does it still function? So we started with our tools, our in vitro tools. So we had mutant podocin cell lines from a patient with a 138Q mutation, compared that to our wild type cells. What's the best way of using pointer? You can see that? Yeah. So what you can see in these cells is that podocin is mislocalized, and it actually co-localized with calnexin, which is an ER marker. So that's in the mutant cells. You can see that you've got mistrafficking to the endoplasmic reticulum. So the idea came about through a discussion with a collaborator in Paris, Alexander Edelman, and he works on cystic fibrosis, so the CFTR molecule. And he found that this interaction with of the mutated CFTR, the common mutation, the F508 deletion, interacts with cytokeratin 8, so a housekeeping molecule. And that, if you disrupt that interaction, that corrects the functional defect in that misfolded protein of CFTR. So we were talking about this, and we thought, well, could the same occur for podocin? So could it be that misfolded podocin also has an interaction with keratin 8, and that's responsible for its mistrafficking? So we used our tools, our cell lines, to have a look at this. So first of all, we looked to see whether keratin 8 associates with the misfolded 138Q podocin, and indeed it does. So by using a couple of methods here, this is the proximity ligation assay, which uses fluorescently tagged oligonucleotides, which you probably can't see very well on the screen, but what you're seeing is if the molecules are close together, you get more red dots, and that's quantified here. So you can see in the mutant cells, you get much more association of keratin 8 with the mutant podocin. And you can do the same by more standard means, by immunoprecipitation, for example. We can show that keratin 8 associates with the mutant podocin, so it pulls down, keratin 8 pulls down the podocin and doesn't pull down wild type podocin. So doing some structural modeling with Richard Sessions in Bristol, he showed that mutation replaces a charged arginine with a neutral residue, and that essentially creates a hydrophobic patch, and that hydrophobic patch is able to then, again, structurally predict and combine keratin. So keratin 8 then binds to that misfolded protein and directs it to a different degradation pathway rather than trafficking normally. And using the molecule that Alexander had used to correct the defect in CFTR, he predicted that molecule sits in that pocket and therefore will disrupt the binding of keratin 8 to the misfolded protein. So we wanted to test whether that molecule can rescue the trafficking of podocin. So this is some of the first experiments that were done. This is using turf microscopy that's only looking at the cell surface. So you can see podocin nicely at the cell. Oops. You can see Podocin nicely at the cell surface. Might be better to use this, so I'll use this so I'm not accidentally moving the slides on. So you can see it nicely at the cell surface in the wild type cells. In the mutant cells, there's no Podocin at the cell surface, and you add this compound and it comes back to the cell surface. So you're seeing nice relocalization of Podocin with the molecule. And that also rescues the function of these cells in vitro. So what we see is normally in wild type cells compared to mutant cells are less adhesive. So they have this cytoskeleton defect, which causes them to be less adhesive. If you add the 407 compound, that significantly rescues the adhesion property. So what it's showing is that you can both rescue the localization of the protein and also rescues the function. So that even though the protein is mutated, it still has function once it gets to the cell surface, which is important. So we wanted to test that in vivo. So we created a mouse model, and this is starting with a mouse that Corinne Antoniak's lab kindly gave us. And what we effectively did, without going through the details, is created a mouse which had the 140Q, which is the mouse equivalent of 138Q on one allele, and a knockout, an inducible knockout on the other allele. So that gives severe disease. We then wanted, We then wanted to treat the mice with 407, so we used a mini pump, and these mini pumps were implanted under the skin, and they delivered the drug for a period of 4 weeks. And what we saw very nicely was actually a very, very significant reversal of proteinuria. So this is over time, you can see up to week 4, and by week 4 in the treated mice, I can't even see my own pointer now. In the treated mice, you're getting proteinuria right back down to baseline levels in this mouse model. And then we can see localization, so the correction of the plasma localization of Podocin. These are mice that haven't been induced, so you should see Podocin nicely on the cell surface there. In the mice with induced disease, that cell surface localization of Podocin disappears. And then you add the drug for 4 weeks, and you've got the plasma membrane localization of Podocin again reappearing. And the histology was remarkable, actually. So complete reversal, complete rescue of histology after 4 weeks. And the ultrastructures of podocyte foot processes was also completely restored. So we saw effectively a complete rescue of this mouse model using the drug. So that's sort of as far as we've got with that particular study. So we think that ERKNet binds misfolded Podocin in the ER and traffics it for proteasomal degradation. If you interrupt that with the small molecule, that rescues the localization, and you get full function back in vitro, but much more convincingly in vivo. So we've done some work towards a medicinal. So this is effectively what's called a tool compound. And what you need is a medicinal chemistry program to optimize the drug-like properties of that compound and make sure that you can get, for example, oral bioavailability, and make it into a compound that has minimal off-target effects, etc. So that's an ongoing program of work. But what I think is quite nice with this is that this has the potential to treat other genetic misfolding diseases. So lots of mutations cause misfolding of proteins. Clearly, this works for the CFTR protein. It works for Podocin. Alexander's group shown it works for alpha-1 antitrypsin. So one of the diseases we want to try is cystinosis, which also has misfolding mutations. So I think it has potential to look at a number of different misfolding proteins by working on that particular mistrafficking biology. So I'm going to switch tack now and talk more about gene therapy. So gene therapy is clearly the other approach that that people have thought about in kidney disease, but so far it's not been achieved, and it's been very successful in a number of other diseases where you can target the cell types or you can target the organs perhaps more easily than the kidney. And I won't go through them all, but you'll know about, for example, treatments for haemophilia B, and there was an approval yesterday for a drug for haemophilia. In the eye, clearly you can get to the eye much more easily than you can to other organs with direct injections. So there's been several successes in treating eye disease and spinal muscular atrophy, dramatic effects in SMA. So that's sort of giving you a flavor of where gene therapy is at the moment in medicine, but it hasn't. Where's the kidney disease? And that's a question a lot of people have asked. So we started a project. So as I say, so far it's been successes mostly in monogenic diseases, in other organs, and the vector is usually adeno-associated virus. So we wanted to ask the question, can we do the same for a monogenic kidney disease? And what are the major hurdles? So the hurdles include that you need to target the correct cell type, and there are at least 32 different cell types in the kidney. And the cargo capacity of AAV, the virus that's used, is relatively limited. So is the gene of a suitable size that it will fit in the payload? And then will it last? Does it stay long enough? And then what kind of dose do you need, and what kind of off-target toxicity is that going to give you? And the approaches that we wanted to use to try and address some of those hurdles include the use of cell-specific promoters, and that's a real advantage of targeting the podocyte, because proteins such as nephrin and podocin are pretty much only expressed in the podocyte, so we have really good cell-specific promoters. Can we get a capsid, an AAV capsid that has tropism for the right cell type? So different AAV serotypes have different tropisms for different cells, different species even. So that's a little bit of a trial and error. Can you get local delivery to the kidney rather than using systemic approaches? And can that gene last for the duration? So this was a PhD project initially for Wen, who was one of my clinical PhD student. And the hypothesis was that podocyte is effectively an ideal target for gene therapy. A, because we have those promoters. B, because it's a terminally differentiated cell that once you get your gene in, it should stay there for life. So, and that's a picture of Wen. So we used AAV as I've described. It's a small virus. It's non-pathogenic. It doesn't integrate, so it's episomal, which is good. It has low immunogenicity, but as I say, it has a relatively small packaging capacity. So what we wanted to do was use AAV as the Trojan horse, as the vector, to transduce the podocyte and express a wild-type version of NPHS2, the podocin gene that encodes our protein. And so Wen made lots of different constructs that fit into the AAV virus. The promoters we used were either human or mouse nephrin promoters. We put podocin in mouse podocin or human podocin, depending on whether we're using human cells or the in vivo mouse experiments, and also GFP tags as a marker protein. And various other constructs are used in the field to try and maximize your ability to express your protein in the cell. And initially we did in vitro experiments. So the in vitro experiments were transduction of our cell lines. So we used podocytes, and as controls we used endothelial cells and proximal tubular cells. And busy slide, but what you can see is that with a CNV promoter, so a generalized promoter, you get nice expression in all the cells. But with a nephrin promoter, you get expression only in the podocytes and not in the other cell types. So we saw nice specificity of expression with the promoter in vitro, so that was a good start. And then we wanted to put the wild type podocin into the mutant podocin cells, so the R138Q cells I've already mentioned. So we did the same thing. We transduced our podocytes. We could see nice expression of the HA tag and of podocin in our cells, and it goes to the right place. And also it's functional. So using the same adhesion assay I've mentioned before, you get normal adhesion in wild type cells, impaired adhesion, less than 50% adhesion in the mutant cells, and then in the cells that have been transduced with wild type podocin, you get a pretty good rescue of adhesion, which is statistically significant. So we're demonstrating we can get it into the cells and it's functional. What about in vivo? So again, we used a podocin knockout mouse. This time it's an inducible knockout of the whole gene. We used either a human or a mouse promoter, so we tried both promoters just in case there was a difference, and we put mouse podocin as our gene of interest into the construct. And we injected the virus via tail vein into these mice, We injected it before induction of disease, so 2 weeks later we induced disease, and then followed them through life up to 16 weeks. And what we see is that you get very significant reduction of proteinuria over that time period. So this is the saline treated mice, and this is quite a big scale. So they get massive proteinuria, and you get very significant attenuation of proteinuria. The two colors just indicate the mouse and the human promoters. And actually, very nicely, you get long-term efficacy here. So the mice that have saline, they're all dead by about 100 days. And the mice that have been treated, about 2/3 of them had survival well beyond a year. So it appears that the effect is long-lived, and we're getting really good efficacy of the drug. And the effects on renal function, the sort of biochemistry. So, for example, cholesterol comes down very nicely. The albumin improves, not yet significance in that small initial group. Urea comes down, and creatinine comes down. So biochemically, they're also improving. And histologically, we can see nice improvement as well. It's not completely normal, but we're getting mostly sort of back to normal ultrastructure and normal histology in the treated mice. And that was assessed blindly by pathologists, so we can see that the number of sclerosed globs, again, the two bars indicate human promoter or mouse promoter. There was no difference between the two. But we're getting nice improvement in sclerosis, and less interstitial fibrosis as well. And we want to understand why is there that little bit of variability? Why are some mice doing better than others? And could that be due to the delivery, the route of delivery, and the fact that it's actually technically can be difficult sometimes to get your tail vein injection just right, et cetera, et cetera. So what we did was firstly look at the level of podocyte transduction in these injected mice, and what we see is there is some variability in transduction, and we're getting about between, say, 15 and 30% transduction of podocytes using the HA tag. So there's variability in transduction in individual mice, and does that correlate to proteinuria? And it seems to. So what we did was we correlated the urinary creatinine.
For this very interesting case. These cases are always so difficult to deal with because of different implications and choosing the right.
Is that a friend of yours, Max? I'll carry on. So with that brief pause. So we looked at urinary albumin creatinine ratio and correlated that with the copies of single-stranded DNA. The virus is single-stranded, so this is by PCR. And even in this, again, relatively small numbers in the first experiment, but we're seeing a significant correlation of proteinuria with the load of virus that you get in the renal cortex. So that seems to suggest that at least some of that variability could be due to the mode of delivery and the success of delivery. So overall, then, we've got a sort of nice proof of principle. The next experiment we did was that was effectively a sort of prophylactic dose, because you're giving your virus before the mice gets proteinuria. So next we did an intervention experiment where we induced the proteinuria. This time, again, this is going back to the R140Q mouse, which I described in the first study. And these get very severe early onset proteinuria. So it's a severe model of disease. So we waited 2 weeks till they get proteinuria, then we gave the intervention, and we followed them through life. So this is the saline mice. What they get is very significant proteinuria. We followed them for a bit longer, and then eventually they get renal failure, and that proteinuria curve comes down. And then with the intervention, we see that initially you get an increase, an uptick in proteinuria, flattens out, and actually comes down much better. I think we're getting better at the IV injections. Same dose of virus, and it comes right down. So your proteinuria comes right down, the albumin level has improved significantly. So that looks very encouraging. Just looking at the histology again, we can see that in the glomerulus, we're getting, using nephrin as a marker, we're getting pretty good transduction efficiency of our podocin into the glomerulus. And this is the histology at the end of treatment. So the untreated mice are sclerosed, and the treated mice have relatively normal histology. So I think that's a really nice proof of concept that we can successfully target the podocyte. Now, with a specific promoter, we can get our protein of interest expressed, and we can get long-term rescue of the disease phenotype. So that's a sort of nice proof of concept that's now led to the spin out of a company called PureSpring. So we were very lucky to get venture capital funding from a company called Syncona that has already spun out 4 other gene therapy companies. So they've got lots of experience. And it's really good to present this in this audience here, actually, because our plan now is to move forward towards first-in-man treatment by 2025. So the timeline that we're coming to. And we started to have discussions already with France and other sort of KOLs in the field about recruiting patients, of getting a registry of patients that will, if you think about patients who are going to go into a trial in 2025, actually those patients, given the natural history of the disease, are probably going to present in the next 2 or 3 years. And those are the patients we need to identify. So we're going to try a sort of global effort, but I think Europe's way ahead of the rest of the world in trying to identify these patients over the next 2 years with really good natural history, prospective and retrospective studies, so that we can really move rapidly into a first-in-man trial with good recruitment. And recruitment is one of the biggest obstacles in rare disease research, as we all know. So just a quick word on the delivery. So clearly in the mouse, we had to give systemic delivery because it's very difficult to get into a mouse renal artery. We try. But the advantages of local delivery, and we're working on a local delivery approach with doing large animal experiments, and I think this is going to be successful, is that
Deixei de conseguir regular o volume porque
you can give both much lower doses, but also and you can also commercially, and this is important, you can get much more virus out of the batches that you make, and that affects the cost of goods hugely. And as we all know, the gene therapies that have been given so far have been very expensive, and this is one of the major drivers, is how much virus you can make from the commercial batches. So this is a very expensive process. These are all things I'm learning as we go along. So a local delivery will mean that you can give less dose, you get less off-target effects. Usually viruses go to the liver preferentially, and you can make more of the product. So just to sort of finish off, then I think this opens a whole exciting new field, certainly for me, who's been working with podocytes for 20+ years. I think, clearly I'm a believer in podocytes, but the podocyte we describe often as a master regulator of glomerular function. And we know it's involved in lots of biological processes, from regulation of filtration via slit diaphragm proteins, maintenance of architecture via the collagens and the basement membrane proteins, regulation of the immune response, inflammatory response within the glomerulus, for example, by regulation of complement, and also metabolic effects. So Richard Coward, who's in Bristol, works has done some groundbreaking work on showing the podocytes an insulin sensitive cell, and is important in the initiation of diabetic nephropathy. So that means that there's lots, if you can target the podocyte correctly, you could go well beyond just the monogenic diseases. So, the monogenic diseases are the obvious ones to start with what I've described, and diseases such as Alport syndrome. But you can also go into targeting inflammatory responses via complement pathway, or the insulin pathways as well for diabetic disease. So I think it opens a whole field of possibility if you can target the cell and target specific pathways. So hopefully I've shown you sort of a couple of ideas for new therapies in nephrotic syndrome, the genetic monogenic diseases in particular, by small molecule approaches for misfolding diseases, but also showing that AAV therapy is feasible and effective in rescuing podocyte genetic mutations and gives you long-term rescue. Lots of people to thank, and I hope I've probably missed people out, but the lab in Bristol, particularly Val and Wen, who did most of those studies I've described, really important collaborators who've helped along the way. Amit Nathwani, who's done the hemophilia trials, really helped us with the capsids, and various funders, and PureSpring as well. So thank you for your attention.
Moin.
Is your friend still online?
I don't know. I don't know who it was. But it was an interesting case. Though. Are there any questions from the room? Is there a question online? There's a question online. Thank you for this very nice talk. Do mice develop an immune response antibody production against human or mouse transgenic NPHP protein? I suppose NPHS or the reporter proteins HAGP over time.
Yeah, so the immune response is a really important aspect, and that is true for all gene therapy approaches so far. And so far, we haven't seen it in the mouse, so we haven't seen any inflammatory response so far and when we've looked in the mouse tissues. But for humans, in human trials, for hemophilia, etc., there have been some. You get neutralizing antibodies to the virus, and there has been some inflammatory responses as well seen. And often patients are treated at least for some period of time with using immunosuppressants for those kind of things. But those are all aspects of getting into man that we have to address and look for very carefully.
I have two questions, if I may. So the first one is about the first part. So did you actually show that the keratin is expressed in podocyte? Because my understanding was that the podocyte is more like a epithelial cell that has converted into a mesenchymal cell that expresses then only mesenchymal intermediate elements. Would they actually express keratin?
Yeah, no, really good question. So wild type podocytes actually express almost no keratinate, but actually the mutant podocytes do. So it seems there must be some kind of feedback loop where your keratinate is expressed when you've got a misfolded protein there. So it's some kind of presumably a misfolded protein response, but we did look at that quite carefully.
Okay. And the other thing is, the other question is about this second part. So I found it very intriguing that you can provide a promoter for a specific cell type, then you get cell-specific expression. So I guess the rationale is that you have transcription factors that are podocyte-specific that recognize the promoter. But aren't there other cells that may have these transcription factors, but the epigenetic changes will close the chromatin, and then these transcription factors will not access the promoters? But when you provide it virally, they might bind it. So did you really not get any extra renal or extra podocyte expression? Did you look?
Yeah. So I can sort of address two aspects of that. One is that one of the aspects of the promoter we've kind of designed a slightly smaller nephrin promoter, but we've looked at what transcription factors may bind, and WT1 is one of the key ones. So we made sure we kept the WT1 binding site, which clearly is relatively cell specific. In terms of off-target expression, I didn't show the data, but we have got data. There is some off-target expression, so it's not perfect. We see some in the liver, but IV, these mice get a big dose of virus to the liver. We saw a little bit in the spleen, but we looked at pancreas, cardiac muscle, etc., etc. So none in most other organs. So there is some, and I think that's something that we have to look carefully at during, as we move up to large animals and clinical trials, what the effect of that might be in terms of off-target toxicity.
Okay, thanks. Thank you. One more in the chat. Are there any techniques on the pipeline for local kidney delivery factors?
Yeah, there is. So we're working on it, and I'm not allowed to tell. This is you find that working with companies that you have to be slightly careful as to when you release data. But we've got good enough data that I think it'll work.
All right. Thank you. Thank you very much. So I guess we move on.